The Cambrian Detonation. In the summer of 1909, paleontologist Charles Walcott was exploring the Burgess Shale Formation in the Canadian Rockies when he discovered something that would shake the foundations of evolutionary biology. What he found wasn't just fossils. It was evidence of an event so rapid and inexplicable that it's been called biology's greatest mystery, the Cambrian Explosion. Approximately 541 million years ago, over a period that geologists estimate at just 5 to 10 million years, an evolutionary eye blink, nearly every major animal body plan that exists today suddenly appeared in the fossil record. Before this moment, life on Earth consisted almost entirely of simple, soft-bodied organisms. After it, the oceans teemed with creatures possessing eyes, armor, articulated limbs, complex nervous systems, and predatory behaviors. What makes this impossible isn't just the speed, but the complexity. These weren't gradual improvements on existing designs. They were revolutionary innovations appearing simultaneously. Trilobites emerged with compound eyes containing thousands of calcite lenses, each perfectly shaped and positioned for optimal vision. Anomalocaris developed sophisticated grasping appendages, and a circular jaw unlike anything before or since. Opabinia appeared with 5 eyes and a flexible proboscis that shows engineering principles of hydraulics and articulation. The mathematical improbability is staggering. Each of these organisms required thousands of coordinated genetic changes to produce their specialized features. Eyes alone demand the simultaneous evolution of light-sensitive proteins, neural processing systems, and sophisticated focusing mechanisms. The standard evolutionary model suggests this should take hundreds of millions of years of gradual refinement, yet it happened in less than 10 million years, perhaps much less. Dr. Douglas Irwin of the Smithsonian Institution studied the genetic changes required for the Cambrian explosion and calculated that the mutation rate would have needed to be at least 40 times faster than anything observed in modern organisms. The probability of so many perfect mutations occurring simultaneously across multiple lineages, he noted in his 2015 paper, approaches statistical impossibility. More disturbing is what the fossils reveal about the complexity of these earliest animals. Recent analysis using electron microscopy has shown that Cambrian arthropods possess neural systems as sophisticated as modern insects. Their compound eyes show the same optical engineering principles found in contemporary dragonflies. Their armor incorporated advanced composite materials that modern materials scientists are only beginning to understand. Perhaps most puzzling is the absence of transitional forms. Darwin himself identified this as a major problem for his theory, writing in On the Origin of Species that the sudden appearance of complex animals in the Cambrian was difficult to explain. He predicted that earlier fossils would eventually be found showing gradual development. Over 150 years of intensive fossil hunting has failed to produce these missing links. Some Cambrian creatures appear to have possessed biological technologies that would require extensive trial and error to develop. The anomalocarids' compound eyes used calcite crystals as lenses, a design that requires precise control over crystal growth and placement. The trilobites' articulated exoskeleton incorporated stress distribution principles that modern engineers recognize as advanced biomechanics. These aren't simple adaptations. They're sophisticated solutions that should require millions of years of gradual refinement. The genetic evidence is equally troubling. Analysis of Hox genes, the master control genes that determine body plans, shows that the genetic toolkit for building complex animals was already fully developed by the Cambrian. But Hox genes are themselves enormously complex, requiring hundreds of coordinated mutations to function properly. Where did this genetic complexity come from? And how did it arise so quickly? Was the Cambrian explosion the result of an environmental trigger we don't understand? Or does it suggest that evolutionary processes can operate far faster than we believe possible? The fossils offer no clear answers, only the unsettling evidence that complex life appeared too quickly to be explained by the gradual accumulation of random mutations. The 3,000-year fish. In 1954, marine biologist Dr. Arthur de Vries was conducting research in the frigid waters of McMurdo Sound, Antarctica, when he encountered something that should have been impossible. He was studying a species of fish called the Antarctic icefish, which thrives in water so cold it should freeze the blood in their veins. What he discovered would challenge everything scientists understood about the pace of evolution. The Antarctic icefish possess a remarkable adaptation. Their blood contains antifreeze glycoproteins that prevent ice crystals from forming, allowing them to survive in waters that reach 2 degrees C. This isn't a simple chemical. It's a complex molecule that requires a specific genetic sequence to produce, and it works through a sophisticated mechanism of binding to ice crystals and preventing their growth. The impossible part is the timeline. Antarctica only glaciated and reached its current frozen state approximately 3,000 to 5,000 years ago. This means the icefish had, at most, 5,000 years to develop a completely new protein with a complex function. According to standard evolutionary theory, the development of a novel protein with a specific function should require millions of years of random mutations and selection. Dr. Chi-Hing Cheng at the University of Illinois conducted genetic analysis of the antifreeze protein genes in 2008 and discovered something even more inexplicable. The antifreeze genes didn't evolve from scratch. They were created through a process called gene duplication and rapid modification. But the modifications required weren't random tweaks. They were precise, coordinated changes that produced a functional protein almost immediately. The antifreeze protein works through a mechanism that requires exact molecular geometry. The protein must bind to specific faces of ice crystals with nanometer precision. If the shape is wrong by even a few atoms, the protein doesn't work. The probability of random mutations producing this exact configuration in just a few thousand years is astronomically low. Even more puzzling is that at least 5 different families of Antarctic fish developed antifreeze proteins independently during this same brief period. Each species evolved its own unique version of the protein, but all of them work through the same fundamental mechanism. This parallel evolution of complex molecular machinery in multiple lineages simultaneously challenges the basic premise that evolution proceeds through random variation. Recent studies using molecular clock analysis suggest the timeline might be even shorter. Dr. William Dietrich at Northeastern University found evidence that some icefish populations developed functional antifreeze proteins in fewer than 1,000 years, perhaps as few as 200 generations. This would require mutation rates hundreds of times faster than observed in any other species. The genetic changes required aren't simple. The antifreeze protein genes show multiple coordinated modifications, including changes to the promoter regions that control when the gene is activated, modifications to the coding sequence that determine the protein's structure, and changes to regulatory elements that control how much protein is produced. All of these changes had to occur in the right order and be preserved by selection, yet there's no evidence of intermediate stages. Laboratory attempts to replicate this evolution have failed. Scientists have tried to evolve antifreeze-like proteins in bacteria using accelerated selection techniques. But even with mutation rates thousands of times higher than natural rates, they haven't been able to produce functional antifreeze proteins in laboratory timescales that correspond to the Antarctic fish timeline. The Antarctic icefish represent a biological impossibility. The evolution of a complex, functionally sophisticated protein system in a time frame that should preclude its development through random mutation and selection. Either evolution can operate far faster than we understand, or something else shaped these fish to survive in their frozen world. The Bombardier's chemical weapon. The Bombardier beetle possesses one of nature's most sophisticated defense mechanisms, and one of evolution's most perplexing mysteries. When threatened, this small insect can fire a boiling hot, toxic chemical spray from specialized chambers in its abdomen with explosive force. What makes this impossible isn't just the complexity of the system, but the fact that it requires multiple perfect components to work simultaneously, and in an intermediate stage would be fatal to the beetle itself. The Bombardier's weapon system works through controlled chemical warfare. The beetle stores 2 chemicals, hydroquinone and hydrogen peroxide, in separate chambers. When threatened, it releases these chemicals into a mixing chamber containing enzymes that catalyze an explosive reaction. The resulting mixture reaches 100 degrees Celsius and is expelled through a steerable nozzle with audible force. The beetle can fire this weapon up to 500 times per second with precise aim. Dr. Thomas Eisner of Cornell University spent decades studying the Bombardier beetle and documented in his 1999 research that the defense system requires at least 5 separate components to function, each of which must be precisely engineered. The beetle needs specialized glands to produce the chemicals, reinforced storage chambers that can contain the reactive compounds, a reaction chamber with heat-resistant lining, enzymes that trigger the explosive reaction at exactly the right moment, and a nozzle system with sphincter muscles for directional control. The evolutionary problem is straightforward. If any single component is missing or malformed, the system doesn't just fail to work, it kills the beetle. Hydroquinone and hydrogen peroxide, when mixed without the proper catalysts and containment, would simply poison the beetle. If the storage chambers aren't properly isolated, the chemicals would react prematurely and cause internal damage. If the reaction chamber isn't heat-resistant, the beetle would be cooked by its own defense mechanism. This creates what biochemist Michael Behe calls irreducible complexity, a system where all components must be present and functional for the system to work at all. Traditional evolutionary theory requires that each intermediate stage provides some survival advantage. But what survival advantage does a beetle get from having explosive chemicals it can't deploy? Or a reaction chamber with nothing to react? Or enzymes that trigger reactions in a beetle without proper containment? Genetic analysis by Dr. Klaus Höldobler in 2004 revealed that the genes controlling the Bombardier beetle's defense system are scattered across multiple chromosomes and require coordinated expression. The beetle's genome contains specialized regulatory sequences that ensure all components of the system develop in the correct order and proportions. This level of genetic coordination suggests a design system rather than a gradual accumulation of random mutations. The fossil record offers no help. Bombardier beetles appear suddenly in fossil deposits from the Cretaceous period, already possessing their complete defense system. There are no intermediate forms showing the gradual development of the chemical weapon system. The earliest Bombardier beetle fossils, dated to approximately 95 million years ago, show the same sophisticated chemical spraying apparatus found in modern species. Laboratory studies have shown just how precisely engineered the system is. The reaction chamber is lined with a resilin-like material that can withstand repeated exposure to 100 degrees C temperatures and explosive pressures. The enzymes that catalyze the reaction are incredibly specific, triggering the explosive reaction only when both chemicals are present in exactly the right ratio. The nozzle system incorporates a muscular valve that can pulse open and closed up to 500 times per second, a frequency that requires specialized muscle fibers not found anywhere else in the beetle's body. Computer modeling by engineers at MIT has shown that the Bombardier beetle's spraying system represents an optimal design solution. The chemical ratios, chamber pressures, and nozzle geometry all conform to engineering principles that maximize defensive effectiveness while minimizing risk to the beetle itself. The system shows design sophistication comparable to modern chemical spray devices. The Bombardier beetle's defense mechanism either evolved through a process radically different from conventional natural selection, or it represents evidence that complex biological systems can appear suddenly without intermediate forms. Either possibility challenges the fundamental assumptions of evolutionary biology. The Octopus Enigma. In 2015, researchers from the University of Chicago and the University of California sequenced the complete genome of the California two-spot octopus. What they discovered was so unexpected that the senior author, Dr. Clifton Ragsdale, told the press, "The octopus genome is unlike any other animal genome we've sequenced. It's as if it was designed by an alien intelligence. The octopus nervous system rivals human complexity with approximately 500 million neurons, comparable to a dog's brain. But here's the impossible part. Octopuses diverged from humans approximately 600 million years ago, long before complex brains evolved in any lineage. This means humans and octopuses developed sophisticated nervous systems completely independently. yet their brains show remarkable functional similarities despite using completely different architectural solutions. What makes this impossible isn't just the convergent evolution of intelligence, but the speed at which it happened. Octopuses only live 1 to 2 years. Their generation time is incredibly short, which should limit the accumulation of complex adaptations. Yet they possess cognitive abilities that rival mammals who've had millions more years and thousands more generations to evolve intelligence. Octopuses can solve puzzles, use tools, plan for the future, and even display individual personalities, all with a brain structure fundamentally different from vertebrate brains. The genetic evidence is even more disturbing. The octopus genome contains 33,000 protein-coding genes, more than humans, who have approximately 20,000. Most strikingly, the genome shows massive expansion of specific gene families that aren't found in any other invertebrates. The protocaterin gene family, which in humans helps wire the brain during development, has expanded to over 168 genes in octopuses, 10 times more than any other invertebrate. Dr. Caroline Albertin, lead author of the genome study, found evidence that octopuses extensively edit their RNA, the molecular messenger that translates genes into proteins. While most organisms occasionally edit RNA, octopuses do it constantly, with over 60% of their neural RNA showing evidence of editing. This allows them to fine-tune protein function in ways that would normally require genetic evolution, potentially accelerating adaptation by orders of magnitude. The octopus eye presents another evolutionary paradox. It evolved completely independently from the vertebrate eye, yet uses the same basic design: a lens, an iris, a retina. More impossibly, octopus eyes actually have better engineering than human eyes. Unlike vertebrate eyes where the optic nerve creates a blind spot, octopus eyes have no blind spot because their retinal wiring is more logical. The probability of two completely independent evolutionary processes arriving at nearly identical sophisticated organs, with the later evolution producing the better design, challenges standard evolutionary thinking. Their camouflage system represents perhaps the greatest mystery. Octopuses can change color and texture to match their surroundings in milliseconds, a feat that requires sophisticated neural control over millions of chromatophores, color-changing cells, in their skin. But here's the impossibility. Octopuses are colorblind. They have only one type of light-sensing opsin in their eyes, meaning they cannot perceive the colors they so perfectly mimic. Recent research by Dr. Desmond Ramirez at UC Santa Barbara discovered that octopus skin contains the same light-sensing proteins found in their eyes. The skin can see independently of the brain, sensing light and color directly, and adjusting camouflage without neural input. This represents a completely novel sensory system that has never been observed in any other organism, a system that would require extensive evolutionary time to develop, yet appeared in a lineage with very short generation times. The octopus's distributed nervous system is equally puzzling. 2/3 of their neurons are in their arms, not their brain. Each arm can operate semi-autonomously, solving problems and manipulating objects without waiting for instructions from the central brain. This requires a level of neural organization that shouldn't be possible without millions of years of refinement. Yet octopuses evolved it in a tiny fraction of the time mammals took to develop centralized nervous systems. Perhaps most disturbing is what genetic analysis reveals about the octopus lineage. Unlike other mollusks which evolve slowly and gradually, octopuses show evidence of rapid genomic reorganization. Their genome has been massively reshuffled compared to their closest relatives, with genes rearranged, duplicated, and modified at rates far exceeding normal evolutionary processes. Some researchers have half-jokingly suggested that octopuses might have extraterrestrial origins. While this is almost certainly not the case, the octopus genome is so unusual and evolves so rapidly that it challenges our understanding of how complex life develops. Either octopuses evolve through mechanisms we don't understand, or they represent an example of accelerated evolution that suggests life can develop sophisticated adaptations far faster than current theory allows. The antibiotic arms race. In 1928, Alexander Fleming discovered penicillin. By the 1940s, it was being mass-produced as a miracle drug. By 1947, bacteria had evolved complete resistance to it. What should have taken millions of years happened in less than 2 decades, and it's happening again with every new antibiotic we develop. The speed of bacterial resistance evolution has shocked microbiologists. Dr. Stuart Levy of Tufts University documented in his landmark studies that bacteria can evolve resistance to novel antibiotics in as little as 2 years, sometimes even during the course of a single patient's treatment. These aren't simple genetic changes. Resistance often requires multiple coordinated mutations that alter cell membrane proteins, modify metabolic pathways, and create entirely new enzyme systems to break down antibiotics. Take MRSA, methicillin-resistant Staphylococcus aureus. Methicillin was introduced in 1959 specifically because bacteria had evolved resistance to penicillin. MRSA appeared in hospitals by 1961, just 2 years later. The genetic changes required weren't trivial. MRSA bacteria possess a completely new protein called PBP2A that standard antibiotics cannot bind to. This protein required multiple precise genetic changes to produce a functional version, yet bacteria across the world evolved it almost simultaneously. Even more impossible is the evolution of NDM1, New Delhi metallobeta-lactamase. This enzyme can break down nearly all known antibiotics, including the last resort drugs doctors use when everything else fails. The gene for NDM1 appeared suddenly in 2008 in India and spread globally within 2 years. Genetic analysis shows it required at least 7 coordinated mutations to create a functional antibiotic-destroying enzyme. 7 perfect mutations, appearing simultaneously, spreading worldwide in months. The mathematical improbability is staggering. Dr. Michael Lynch at Indiana University calculated that the probability of multiple coordinated mutations occurring spontaneously in a single bacterium is approximately 1 in 10^15 for each additional required mutation. Yet bacteria are producing these exact multi-mutation combinations in time frames measured in years, not millions of years. What makes this truly impossible is that bacteria seem to evolve resistance to antibiotics they have never encountered before. In 2012, researchers discovered bacteria in isolated caves that had been sealed for over 4 million years. When tested, these bacteria, which had never been exposed to modern antibiotics, already possessed genes that conferred resistance to multiple modern drugs, including synthetic antibiotics that don't exist in nature. They had resistance to chemicals that hadn't been invented yet. Dr. Jerry Wright at McMaster University found that antibiotic resistance genes are far older than antibiotics themselves. The genetic machinery for destroying penicillin existed in bacteria long before Fleming discovered the drug. It's as if bacteria were prepared in advance for chemical weapons that wouldn't exist for millions of years. The mechanism of rapid resistance evolution is equally mysterious. Bacteria can transfer resistance genes between species through horizontal gene transfer, essentially sharing genetic code like files on a network. But this requires sophisticated cellular machinery for cutting, transferring, and integrating foreign DNA. The probability of this system evolving through random mutation is vanishingly small, yet it's ubiquitous in bacteria. Recent studies have revealed something even more troubling. When bacteria are exposed to sublethal doses of antibiotics, they don't just randomly mutate until resistance appears. They activate specific genetic programs that increase mutation rates in targeted regions of their genome, regions that code for cell membrane proteins and metabolic enzymes that interact with antibiotics. It's as if bacteria have a built-in system for accelerated directed evolution that activates when needed. Dr. Susan Rosenberg at Baylor College of Medicine discovered that stressed bacteria activate error-prone DNA repair systems that deliberately introduce mutations at rates thousands of times higher than normal, but only in genes relevant to the current stress. This isn't random evolution. It's targeted genetic modification that produces solutions to environmental challenges at speeds that shouldn't be possible through chance mutation alone. The implications are profound. Bacteria are evolving complex solutions to novel challenges in real time, producing sophisticated multi-component resistance mechanisms in years rather than millions of years. Either microbial evolution operates through mechanisms radically different from what we understand, or bacteria possess systems for accelerated adaptation that suggest evolutionary processes can be directed and controlled rather than purely random. The antibiotic resistance crisis isn't just a medical problem. It's evidence that evolution can proceed at rates that challenge the fundamental assumptions of evolutionary biology. The Hawaiian Radiation. The Hawaiian Islands are young. The oldest island, Kauai, is only about 5 million years old. Yet these islands host one of the most spectacular examples of rapid speciation ever documented. The Hawaiian Silver Sword Alliance. From a single ancestral plant species that arrived on the islands, more than 50 distinct species evolved, each adapted to radically different environments from high-altitude volcanic deserts to tropical rainforests. The impossible part. This all happened in less than 5 million years. Dr. Gerald Carr of the University of Hawaii studied the Silver Sword Alliance and discovered that these plants show more morphological diversity than many plant families that have been evolving for 100 million years. Some silver swords are tiny alpine shrubs growing in volcanic cinder at 13,000 feet elevation. Others are tree-sized plants growing in rainforests at sea level. Some have sword-like leaves covered in reflective silver hairs. Others have soft, broad leaves adapted to wet environments. Yet genetic analysis proves they're all descended from a single tarweed ancestor that arrived from California less than 5 million years ago. The speed of diversification is staggering. To produce this level of morphological and ecological diversity through standard evolutionary processes should require at least 50 million years based on speciation rates observed in other plant families. The silver swords did it in 1/10 the time. Each species shows sophisticated adaptations that should require millions of years of gradual refinement. Specialized root systems for extracting water from volcanic rock, modified leaf structures for reflecting intense UV radiation, altered flowering mechanisms for attracting specific pollinators found only in Hawaii. Even more impossible is the convergent evolution within the group. Multiple silver sword species independently evolved similar adaptations in response to similar environments. At least 3 separate species evolved the characteristic silver leaves independently, each using different genetic mechanisms to produce the same effect. This suggests that evolution isn't proceeding through random mutation, but somehow finding optimal solutions repeatedly through unknown mechanisms. The genetic analysis reveals something even stranger. Dr. Bruce Baldwin at UC Berkeley sequenced the genomes of multiple silver sword species and found that key adaptive genes show evidence of rapid targeted modification. Genes controlling leaf shape, flowering time, and drought tolerance show mutation rates 10-50 times higher than neutral genes, as if evolution was somehow focused on the genes that needed to change while leaving other parts of the genome relatively stable. What makes the Hawaiian silver swords impossible isn't just the speed of their diversification, but the complexity of the adaptations they evolved. These aren't simple changes. They're sophisticated solutions to challenging environments that require coordinated changes across multiple genetic systems. The alpine silver swords, for example, needed to evolve freeze tolerance, UV protection, water conservation mechanisms, and specialized root systems all simultaneously to survive at high elevation. Any one of these adaptations without the others would be lethal. The pattern repeats across Hawaiian ecosystems. Hawaiian honey creepers, a group of birds, evolved into over 50 species from a single finch ancestor in less than 4 million years. Hawaiian Drosophila flies diversified into over 800 species in roughly the same time frame. Hawaiian land snails radiated into over 750 species. In each case, the rate of speciation and adaptive evolution is 10 to 100 times faster than the rates observed in comparable lineages on continents. Something about island environments seems to accelerate evolution to impossible speeds. Either natural selection operates far more efficiently in isolated populations than current theory suggests, or these rapid radiations are evidence of evolutionary mechanisms that we don't yet understand. The Hawaiian silver swords stand as living proof that complex adaptations can evolve in tiny fractions of the time that evolutionary theory predicts should be necessary. They challenge the gradualist view of evolution and suggest that life can transform itself at speeds that border on the impossible. The Cichlid Explosion. In East Africa's Rift Valley, 3 Great Lakes contain one of evolution's most impossible achievements: the cichlid species flocks. Lake Victoria alone hosts over 500 species of cichlid fish, each with unique feeding behaviors, body shapes, and ecological niches. The impossible part: the lake is only 15,000 years old. The entire Cichlid Radiation, 500 species with sophisticated specializations, evolved in 15,000 years or less. Dr. Axel Meyer of the University of Constance has spent decades studying African cichlids and describes their evolution as the fastest rate of large-scale speciation ever documented. To put this in perspective, 500 species in 15,000 years means a new species evolving every 30 years on average. In human terms, that would be like your grandfather belonging to a different species than you. The diversity is staggering. Some Lake Victoria cichlids have evolved to scrape algae from rocks with specialized rasping teeth. Others have developed crushing jaws for eating snails. Still others have elongated jaws for sucking insects out of crevices. There are cichlids with teeth on their tongues for pulling scales off other fish, and cichlids with protrusible mouths for plucking eyes from prey. Each of these feeding specializations requires dozens of coordinated changes to skull morphology, muscle attachments, and neural control systems. The genetic basis for this rapid evolution is even more puzzling. Dr. Richard Durbin's team at the Sanger Institute sequenced the genomes of 60 cichlid species in 2014 and discovered something impossible. Despite their dramatic morphological differences, the cichlids show remarkably little genetic divergence. Species that look completely different and occupy completely different ecological niches are virtually identical at the DNA level, typically differing by less than 0.5% of their genome. This violates the basic principle that morphological evolution requires genetic evolution. Somehow, cichlids are generating enormous phenotypic diversity from minimal genetic change. Dr. Emily Darren at Stanford found that cichlids show exceptionally high rates of regulatory mutation, changes not in the genes themselves, but in the control switches that determine when and where genes are activated. A single regulatory mutation can cause a gene to be expressed in a different part of the developing fish, leading to dramatic changes in form without requiring changes to the gene itself. But even regulatory evolution shouldn't happen this fast. Laboratory experiments on fish evolution typically show that producing even simple morphological changes requires hundreds of generations of strong selection. The cichlids are producing complex, integrated changes in mere thousands of generations, changes that should be impossible without significantly more time. The Lake Victoria cichlid flock faces an even greater mystery. Approximately 14,500 years ago, the lake completely dried up during a severe drought. When it refilled, it was repopulated by just a handful of cichlid species from nearby rivers. The current 500-plus species all evolved from those few founders in approximately 14,500 years, and possibly much less, since environmental evidence suggests the lake didn't reach its current size and stability until about 12,000 years ago. Dr. Ole Zeehausen at the University of Bern discovered that many Lake Victoria cichlids show evidence of hybrid ancestry. They formed when different species interbred. This suggests that cichlid diversification isn't just occurring through standard speciation and mutation, but through recombination of existing genetic variation in novel ways. Hybrid speciation can potentially accelerate evolution by combining adaptive genes from different lineages, but it still requires that those adaptive genes existed in the ancestral population. The cichlid story becomes more impossible when we examine other lakes. Lake Malawi contains over 850 cichlid species that evolved in less than 2 million years. Lake Tanganyika holds over 250 species that emerged in approximately 9 or 12 million years. In each case, the rate of speciation is orders of magnitude faster than observed in any continental environment. Most troubling is the convergent evolution between lakes. cichlids in all three lakes independently evolved similar body forms and feeding specializations. Each lake has algae scrapers, snail crushers, insect pickers, and scale eaters. Yet these forms evolved completely independently in each lake from different ancestral species. The probability of evolution producing the same sophisticated solutions multiple times through random mutation is vanishingly small. The African cichlids represent an evolutionary impossibility. Hundreds of species with complex, integrated adaptations, evolving in timeframes that are 100 times too short according to conventional evolutionary theory. Either evolution can proceed through mechanisms that generate adaptive change far faster than random mutation and selection, or the cichlids possess some form of evolutionary acceleration system that we don't yet understand. The human brain paradox. The human brain contains approximately 86 billion neurons and 100 trillion synaptic connections. It's the most complex structure in the known universe, and according to the fossil record, it evolved from a chimp-like ancestor to its current size and complexity in less than 6 million years. The problem, the mathematical probability of evolving a structure this complex in this time frame, is essentially zero. Dr. Susanna Herculano Housel at Vanderbilt University has mapped the neuron counts across primate evolution and found something troubling. The human brain didn't just get bigger. It underwent massive reorganization. Our prefrontal cortex, the region responsible for abstract thought and planning, is approximately 6 times larger than expected for a primate of our body size. The neural connections in this region show architectural sophistication that has no precedent in the primate lineage. The genetic changes required are staggering. Dr. David Kingsley's team at Stanford identified approximately 2,000 genetic regions that show evidence of accelerated evolution in humans after our split from chimpanzees. Many of these rapidly evolving regions control brain development. But here's the impossible part. Most of these regions aren't genes at all. They're regulatory elements that control when and where genes are activated during development. Regulatory evolution is supposed to be slow and gradual because most regulatory changes are harmful. Yet humans show evidence of positive selection on dozens of regulatory elements controlling brain development, all occurring simultaneously across a timeline of just 6 million years. Each beneficial regulatory mutation needs to occur, be preserved by selection, and spread through the population before the next one can be added. The standard model suggests this should take hundreds of millions of years for the number of changes we see. The brain's energy requirements present another impossible hurdle. The human brain uses 20% of the body's energy despite being only 2% of body weight. This means that as brains were evolving larger, our ancestors needed to simultaneously evolve more efficient metabolisms, better thermoregulation, and enhanced cardiovascular systems to support the increased energy demands. All of these changes needed to occur in coordination. A brain that consumes more energy without the metabolic systems to support it would be lethal. Dr. Bruce Lahn at the University of Chicago discovered 2 genes, ASPM and microcephalin, that show clear evidence of recent positive selection in humans. Both genes regulate brain size during development. Mutations in these genes cause microcephaly, a condition where the brain is dramatically smaller than normal. The fact that these genes were under intense selection pressure in recent human evolution suggests that brain enlargement was happening far faster than neutral drift would allow. Even more puzzling is the fossil evidence. The transition from Australopithecus, brain size 50 cc, to modern Homo sapiens, brain size 350 cc, occurred over approximately 4 million years. This means the brain was tripling in size, not through overall body growth, but through specific enlargement of neural tissue. The metabolic cost of this expansion would have been enormous, requiring coordinated changes across multiple biological systems simultaneously. The archaeological record shows something equally mysterious. The material culture of our ancestors remained remarkably simple for millions of years, then suddenly exploded in complexity around 70,000 years ago. This suggests that the physical evolution of the brain preceded its full cognitive utilization by millions of years. Why would evolution produce a brain capable of abstract mathematics, symbolic language, and complex technology millions of years before these abilities provided any survival advantage. Recent genetic studies have identified that many human brain genes show signatures of introgression, genetic material acquired through interbreeding with other human species like Neanderthals and Denisovans. Dr. Joshua Akey at Princeton found that some of these introgressed genes affect brain development and neural function. This suggests that human cognitive evolution wasn't just the result of gradual selection within a single lineage, but involved genetic exchange between multiple human species, potentially accelerating evolution by combining adaptive variants that had evolved independently. The human brain represents an evolutionary impossibility, a structure of staggering complexity that evolved at speeds far exceeding what population genetics predicts is possible through random mutation and selection. Either human brain evolution involved mechanisms beyond standard natural selection, or it benefited from factors that dramatically accelerated adaptive change in ways we don't yet understand. The molecular clock paradox. One of molecular biology's fundamental tools is the molecular clock, the principle that genes mutate at relatively constant rates over time, allowing scientists to date evolutionary events by comparing genetic differences. But when scientists applied this tool to certain organisms, they discovered something impossible. Species that should be separated by hundreds of millions of years, according to the fossil record, show genetic similarities, suggesting they diverged just millions of years ago. Dr. Simon Ho at the University of Sydney has documented dozens of cases where molecular clocks give dates that are wildly inconsistent with fossil evidence. His 2014 paper describes what he calls time-dependent rate variation, the observation that mutation rates appear to change over time, generally slowing down over longer time scales. But this creates an impossible paradox. If mutation rates were faster in the recent past, then older evolutionary events would require even less time than already impossible timelines suggest. The most dramatic example involves birds. The fossil record clearly shows that modern bird orders diversified after the extinction of the dinosaurs 66 million years ago. But molecular clock analysis of bird genomes suggests this diversification happened in just 10 out of 15 million years. The genetic differences between bird orders are so small that standard mutation rates would place their divergence far more recently than the fossils allow. Dr. Richard Buggs at Queen Mary University of London discovered an even more troubling pattern. When comparing living fossil species, organisms that have remained morphologically unchanged for millions of years, their genomes show the expected amount of genetic change for their supposed age. But when comparing rapidly evolving lineages, the genetic changes seem too small for the morphological differences observed. It's as if morphological evolution and genetic evolution are somehow uncoupled. The tardigrade genome presents perhaps the greatest paradox. These microscopic creatures can survive extreme conditions, including the vacuum of space, extreme radiation, and freezing temperatures. Genetic analysis suggests they acquired many of these survival abilities through horizontal gene transfer, taking genes from bacteria, fungi, and other organisms and incorporating them into their own genome. This alone is remarkable, but the impossibility is this. Tardigrades show evidence of acquiring over 6,000 foreign genes, approximately 17% of their genome, over a period estimated at just 50 million years. Horizontal gene transfer is supposed to be rare in complex organisms. The probability of successfully integrating a foreign gene into a functioning genome is extremely low. Most gene transfer events are harmful or neutral. Yet tardigrades have successfully integrated thousands of foreign genes, each requiring multiple coordinated changes to regulatory elements, promoter sequences, and control systems. The probability of this happening through chance is essentially zero. Dr. Bob Goldstein at the University of North Carolina sequenced tardigrade genomes and found that the foreign genes aren't randomly distributed. They're clustered in regions of the genome associated with stress response. It's as if the tardigrade genome was somehow actively targeting and acquiring genes that would help it survive extreme conditions. This suggests a directed process rather than random horizontal gene transfer. The The molecular clock paradox extends to mutation rates themselves. When scientists measure mutation rates directly by sequencing parent and offspring genomes, they get rates that are 2 to 10 times higher than the rates calculated from comparing species over longer time scales. This means either mutation rates are slowing down over evolutionary time, which makes no biological sense, or many mutations are being removed from populations far faster than standard evolutionary models predict. This creates an impossible timeline problem. If actual mutation rates are higher than evolutionary calculations assume, then the amount of time required for observed genetic changes is even shorter than already impossibly brief estimates. The cichlids that evolved in 15,000 years would have needed even less time. The human brain that evolved in 6 million years might have evolved in just 2 to 3 million years. Every evolutionary timeline becomes even more compressed and even less plausible. The molecular clock paradox suggests that either our understanding of mutation rates is fundamentally wrong, or evolutionary change can proceed through mechanisms that generate adaptive mutations far faster than random processes should allow. Either possibility challenges the foundation of evolutionary biology. The quantum evolution's question. In 2016, physicists at the University of Vienna demonstrated something that should be impossible: quantum coherence in biological molecules at room temperature. Quantum effects were thought to exist only in the ultra-cold, isolated conditions of physics laboratories. But they found quantum coherence in photosynthetic proteins and suggested it might be widespread in biological systems. The implications for evolutionary theory are profound and disturbing. Dr. Gregory Scholes at Princeton discovered that plants use quantum mechanics to enhance photosynthesis efficiency. Light harvesting proteins in plant cells maintain quantum coherence, allowing energy to simultaneously explore multiple pathways and find the most efficient route to reaction centers. This isn't just chemical efficiency. It's using quantum superposition to solve complex optimization problems in real time. The impossible part: quantum coherence is extremely fragile and collapses almost instantly when exposed to thermal noise and molecular vibrations. Yet, plants have evolved molecular structures that protect quantum coherence at room temperature, something physicists thought was fundamentally impossible. The protein structures required to shield quantum effects from thermal noise are incredibly precise, requiring exact positioning of atoms to within a fraction of a nanometer. How did plants evolve such precise molecular engineering? Random mutations cannot feel quantum effects. Natural selection can only select for phenotypes, observable traits that affect survival and reproduction. But quantum coherence doesn't affect the observable phenotype directly. It just makes energy transfer slightly more efficient. The selective advantage of going from 95% efficiency to 97% efficiency through quantum mechanics would be so small that genetic drift would overwhelm selection. Yet the quantum coherence mechanisms in plants show clear evidence of optimization. The protein structures aren't just barely functional, they're near-optimal solutions to the physics problem of maintaining quantum effects in warm, wet, biological environments. Dr. Elisabetta Collini at the University of Padova found that photosynthetic proteins show design features that suggest engineering, symmetry breaking that prevents quantum decoherence, precisely tuned molecular vibrations that enhance quantum transport, and protective protein scaffolds that shield quantum processes from environmental noise. The implications extend beyond photosynthesis. In 2011, Dr. Luca Turin at MIT proposed that the human sense of smell might use quantum mechanics to distinguish molecular shapes. Olfactory receptors might be detecting molecular vibrations through quantum tunneling of electrons, a process that requires the same kind of precise molecular engineering found in plant photosynthesis. If biological systems regularly use quantum effects, then evolution faces an impossible optimization problem. It must tune molecular structures to sub-nanometer precision to enable quantum coherence. But the selective advantages of quantum optimization are too small to drive selection, and random mutations cannot know that specific molecular geometries will enable quantum effects. Dr. Jim Al-Khalili at the University of Surrey has proposed that quantum effects might actually enhance evolutionary processes themselves. If quantum tunneling allows molecules to cross energy barriers they couldn't cross classically, then chemical reactions in cells, including DNA replication and protein folding, might have access to outcomes that would be impossible through classical chemistry alone. This could potentially accelerate evolution by allowing biological systems to explore molecular configurations that would be unreachable through random mutation. This leads to a disturbing possibility. What if evolution isn't entirely random? What if quantum effects in biological molecules allow living systems to explore solution spaces more efficiently than pure randomness would allow? This wouldn't be intelligent design, but it would be directed evolution, a process where quantum mechanics guides the exploration of genetic space toward functional solutions, faster than classical random mutation could achieve. Recent experiments on quantum biology have suggested something even more radical. Dr. Matthew Fisher at UC Santa Barbara has proposed that quantum entanglement might persist in neural phosphorus atoms, potentially affecting brain function. If quantum effects are common in biology, then the distinction between random mutation and directed adaptation becomes blurred. Quantum mechanics allows systems to explore multiple possibilities simultaneously before collapsing into the most favorable outcome. Could quantum effects explain how species evolve complex adaptations in impossibly short time frames? Could organisms be using quantum processes to accelerate adaptation by simultaneously exploring multiple genetic configurations before settling on successful ones? The evidence is circumstantial but growing. Biological systems show signatures of quantum optimization in their molecular structures, and quantum effects appear far more common in living systems than classical biology assumed. If evolution incorporates quantum processes, then the timelines that seem impossible under classical evolutionary theory might actually be plausible. Quantum-assisted evolution could potentially generate complex adaptations, orders of magnitude faster than random classical mutation. This would explain why Cichlid speciate at rates 100 times faster than expected, why bacteria evolve resistance to novel antibiotics in years, and why the Cambrian explosion produced complex body plans in a geological instant. The quantum evolution question remains open, but it suggests that life might operate through principles that transcend the random, gradual processes of classical evolutionary theory. These cases represent some of the most compelling challenges to the standard evolutionary timeline. From the explosive diversification of the Cambrian period to bacteria that evolve complex resistance in years rather than millennia, from fish that develop sophisticated biochemistry in thousands of generations to plants that engineer quantum coherence in their proteins, the evidence suggests that evolution can proceed far faster than classical theory predicts. What unites these cases isn't a rejection of evolution itself, but a question about its mechanisms and speed. The fossil record, genetic evidence, and laboratory observations all confirm that species change over time. But the rates of change we observe, particularly for complex integrated adaptations, seem to require either mechanisms beyond random mutation and selection, or timelines that are fundamentally wrong. Whether these rapid evolutionary leaps are evidence of quantum-assisted optimization, horizontal gene transfer, regulatory shortcuts, or processes we haven't yet discovered, they remind us that life on Earth is more mysterious and more adaptable than our current theories can fully explain. Evolution, it seems, has access to shortcuts we don't yet understand. Shortcuts that allow complex life to transform itself at speeds that should be impossible.